Hello, thank you for joining me this morning for this virtual session. I'm going to be talking for a few minutes about research that has been ongoing in my laboratory and with colleagues around the world on transboundary One Health research on alien invasive species, specifically in the Americas. And as we look through this, I want to point out that I'm going to be covering more than 30 years in about 10 minutes. So I'm going to move very quickly through slides. And if you can't see everything in detail, just sit back and listen to the main thread of the story. And of course, this will be available online, both through the conference and also I will be posting it online so that you can look at details later. So in the last over 30 years, we have been doing work specifically on invasive species that are either vectors or agents of various types of infectious diseases. The first we started with this was on the boundary of Canada and the United States, specifically New York State and Ontario, along the St. Lawrence River. And this was in 1989 when we were first experiencing the invasion of North America from Europe by zebra mussels and quagga mussels. And so being someone in the area that had some expertise in mollusks and other things, I was asked by local people, both in the Canada and the US, to develop a task force to learn to understand what the mussels were doing, what kind of impact they were going to have on us, and how we might better prepare for them. You can see on the map that, as we know, the Dreissenids now are throughout North America, but specifically they have moved down the St. Lawrence Seaway into other river systems, so they have been a transboundary issue from the very outset. In the Lake Ontario drainage basin, we have primarily focused on the St. Lawrence River itself, the upper river, almost down to Montreal and then all the way to Kingston. We've also looked, though, at the northern part of Lake Ontario, from Toronto across to the east, and also the more shallow, more agricultural area around Kingston. With these organisms, with these mussels, we have used a lot of techniques to collect them and to study them, but the most important monitoring method we have developed is the use of navigational buoys that are in the seaway and used for navigation of large ships into and out of the major harbors. This is a really powerful technique because by using these as monitoring platforms, we can actually go all the way from the surface of the water down to the very bottom of the river or the lake on the anchor. And in addition to that, sampling that entire profile, we can see that each of these navigational buoys, of course, goes in the same exact spot year in and year out. It has done so for more than the last half century. And each one has a sign on it that tells you exactly what that buoy was located. So it makes for a very good way of monitoring distribution. Another thing is that they pull these buoys out of the seaway during the winter time and clean them and so on. And so we can simply go up to them on land during the winter. And you can see over here on the right, this is my wife that's examining them with me. You can see all of the little black marks, all of those are mussels. And so we can simply scrape the mussels off from there and do whatever we want to do with them in terms of examination. Well, the primary thing we wanted to examine them for, besides monitoring the movement of the mussels into the system, is we wanted to start using them as sentinel monitors for determining the distribution of human infectious and animal infectious pathogens. Specifically, we've looked at Cryptosporidium, Giardia, and some of the human and animal infectious Microsporidia. And we are using these because the mussels, being bivalves, they are filter feeders. And as they filter the water, they concentrate whatever is in the water in a particular matter, and that includes these pathogens. So we pioneered this work of using these bivalves as sentinel monitors. We take them off of the buoys, and once we've gotten them from the buoys, we take them in the laboratory and use a variety of techniques, from standard histological techniques on the left to molecular techniques on the right for very specific and very accurate identification of various pathogens. And then we go back to the places where we collect them, and we couple what we found about the infection by these pathogens with the distribution of the buoys. And here you can see a profile extending all the way from the westernmost part of Lake Ontario across through Toronto, Kingston, various towns in New York, and then all the way almost to Montreal. And you can see the total number of pathogens found in the mussels at each buoy. So you get a good span of what is out there in its relative abundance. You can also narrow it down further, and we've done that in specific location. This just in Lake Ontario. You can see over on the left, very little was found around Toronto in this particular year, a lot more down toward Kingston. And so we can get an idea of how these organisms are distributed, and therefore where they likely are coming from, and whose water supply they're likely to contaminate. And so it has become quite a powerful method. Besides the Dracaenas, though, we have other invasive species that we're using as well. We've begun to use Corbicula fluminea from Asia a lot. Now, Corbicula fluminea is more of a southern species, but it's been around a lot longer, having invaded in the early parts of the 1900s. But more recently, it has made its way also into the St. Lawrence system. And in fact, we have been doing more transboundary research there with Tony Riccardi and his group at McGill University, where we've actually been trying to understand the movement of Corbicula from warmer into colder waters by looking at cold tolerance. And we just published this paper a couple of months ago, actually, showing that, in fact, variations in cold tolerance will affect the ability of these mussels to move into different habitats and to adapt. Another thing that we are doing is we're finding Corbicula like Dracaena is also very effective as a sentinel biomonitor. Not only are these biomonitors, but they can also be hosts for dangerous trematodes. Mollusks, of course, are the intermediate host for most trematodes, and not only bivalves, but especially the gastropods. And so we did more work on a transboundary level with an international team between Canada and the US, looking at invasive gastropods and bivalves, and have found that they do, in fact, host, once they get into the new habitat here in North America from various countries, various continents, they do carry our local parasites. And those parasites have been shown to cause outbreaks of disease in wildlife, wild birds, as well as domesticated animals. But when it comes to disease spread, one of the most important things is vector mosquitoes and other insects. We've been looking at mosquitoes very carefully now along several transects in a big program that we have called From the Arctic to the Equator. And we've teamed with groups in many countries. We work across the various borders of our countries, and we have defined transects that we explore so that we can get an idea of mosquitoes in general, but especially invasive mosquitoes that carry disease. Get an idea of how their distribution relates to climate, topography, and geography. The primary transect we've looked at so far covers many different nations. So you talk about transboundary, we have many boundaries here because all of the islands of the Caribbean are, of course, most of them are different countries. We also have South America and North America here, the United States, Venezuela, Colombia. This has been a corridor for movement of disease and vectors of disease for several centuries, actually, especially with diseases like yellow fever, transmitted primarily by Aedes aegypti, an invading mosquito from Egypt. But in the last 10 years, we've gotten two serious wake-up calls, one from chikungunya virus and another one from Zika virus, also spread by the same mosquitoes, the same invasive mosquitoes. And they also have moved back and forth between North and South America along this Caribbean corridor. We have established several monitoring sites throughout the Caribbean, but also in South Florida, in Georgia, all the way up to through the coastal plain, through the Piedmont, into the foothills, and to the top of the Cumberland Plateau, which is the first major altitudinal barrier to movement and colonization by mosquitoes. And so we're looking at this transect in terms of, again, of understanding how these vectors may be distributed and therefore how the diseases they carry may be distributed. We collect the mosquitoes, we take them back to the laboratory and identify them microscopically as well as through PCR analysis. Here you can see a variety of traps that we use out in the field. Besides the traps, though, we also go into small surveys, short-term surveys using handheld microscopes, as well as basting bulbs to take water samples out of bromeliads and out of small water pockets here and there. And we also use human landing kits where we basically just take vials, and when the mosquito lands on us, we hold it up, we collect the mosquito, take it back to the lab. Using these techniques, we have made a lot of important finds in St. Kitts, right in the most heavily trafficked area of the city of Basseterre. We have found large numbers of Aedes aegypti larvae in flower pots. In San Juan, Puerto Rico, the flower pots are not such a big problem. They drain them well, but they have failed to look at the water meter wells, which accumulate water from rain, and those are full of Aedes aegypti. In the British Virgin Islands, we found that large truck tires that they put out to serve as places to sit for pedestrians are actually filled with Aedes aegypti. And in the very nice little boutiques and in the nice little cafes along the sidewalks, you have Aedes aegypti breeding in decorative fountains. Even when you get up into Florida, South Florida, a very opulent area, a lot of nice boutiques and shops and high-end facilities there, you still get Aedes aegypti in very large numbers. And you also begin to get Aedes albopictus at this point. As you move farther north into the Appalachian foothills, then you get a lot of native mosquitoes that we've been collecting, but Aedes albopictus dominates in that more northerly environment and a little bit higher altitude, and you have completely lost Aedes aegypti by that time. And then when you jump up to Montego, Tennessee, at the top of the Cumberland Plateau, still no Aedes aegypti. Aedes albopictus is still a very important species there, but also Aedes japonicus, another alien invasive species. Well, we started a new transect this summer, just 2 months ago. We began to do work in our old haunts along the St. Lawrence River. And here you can see a number of sites that we have established and collected mosquitoes in the northern Adirondacks and the St. Lawrence River Valley. And while we have not found any of these exotic invasive species in that habitat, we are now in this much more northerly environment. This is actually, by the way, right here on this star, this is exactly halfway between the equator and the North Pole. At this point, we have found things like Aedes intrudens, which, while it is not an exotic invasive, it is a species that is aggressively spreading throughout much of North America. So this just gives you an overview of the kinds of things we've been up to. It all involves transboundary work. It all involves working together with other countries, other industries, other sectors, and so on. And I will invite you now to just ask questions here or to contact me by email or in any other way so that I can continue to share with you. Thank you for attending.